Thanks a lot, Sarka, Emma, Miriam, and Swash for putting up a nice show. I see many of my old acquaintances, particularly my dear friend Thomas. So a big hi to Thomas and all whom I know. So it's great to have an invitation from this forum, which is strictly confined to the intermediate filaments, and we are hearing a lot of these advancements in these intermediate filaments from these different talks. So my lab actually works on lamin A, so principally lamins, but over the past decade, since I set up my lab, I have been focusing on lamin A, and these are the two figures which are my favorite. One is the paracrystal from the transmission electron microscope from my postdoc days at Bob's lab, Professor Goldman's lab at Northwestern, and the other one is the image from my lab. This is a super resolution image of lamin A and B in C2C12 cells, which are absolutely fascinating. So I'm really fascinated with this, with the lamins, and let's see. Let's move. Okay, so first of all, I need to thank all my my former graduate students, Abhinanda, Pitha, Manindra, and Shubhana have already done their PhD with me and are placed at different places in the US and Europe. Chandrayee and Duhita will be the stars today who will be presenting their talks. Shreyashi and Shubhradeep, they are still rookies and working on the different aspects of the lamin A, which I cannot discuss today for the paucity of time. And so basically, we all know about the architecture of the nuclear lamina, which started with this brilliant finding from Uli Abe's lab, and this where this electron, the transmission electron micrograph of the Xenopus oocyte showed this wonderful network of mainly orthogonal arrangement, which was kind of modified and corrected by Goldberg in 2008, where it was shown that it's actually a combination of both the orthogonal filaments overlaid by multiple parallel fibers of lamin A. And the most revolutionizing science came, I think, in the recent times from Ohad's lab, where the entire paradigm of the intermediate filament has been changed, because now we move from the paradigm of, let's say, 10 nanometer or more than 10 nanometer to 3.54 nanometer thickness, which actually is an absolutely fascinating new invention in the field. So just to give a little introduction, I think all of you are pretty much familiar with the lamins, which is a nuclear intermediate filament. Well, we still continue to call it an intermediate filament as per the nomenclature. So they are present inside the nucleus, and the mechanic. I'm just wondering whether there is a pointer. There should be a pointer. Yeah, got it. Yeah. So when we have the cell on this extracellular matrix, the cell experiences various mechanical cues which are transmitted to the nucleus via the cytoskeleton, and this is actually magnified here. And the cytoskeleton is composed of intermediate filament, the actin here, and the microtubule, and this all terminates into the nucleus, where they are connected to the lamins, which are the nuclear intermediate filaments, which play a lot of functions in nuclear homeostasis, starting from transcriptional regulation, replication, DNA damage repair, maintenance of the chromosome architecture, and ever expanding fields. So what is fascinating, that these mechanical cues are modified under the different conditions the cells are exposed to. So it can be the case of cancer cell, which is metastasizing through the interstitial spaces, where the nucleus gets squeezed, and we have seen wonderful papers from Jan Lammerding's lab also, about how the nuclear remodeling constantly takes place with the remodeling of the lamin. And also it can be the muscle cells. And we are interested mainly in these two types of research. And this is what my lab has been doing for the past decade. When I began my lab, I started understanding the viscoelasticity of the lamins. As we all know, that the lamins are the biological fibers, and they have viscous and an elastic component. And the viscoelastic component of lamin was shown first by our group. And the first report from the intermediate filament was of lamin B1. And the viscoelastic effect of lamin A mutations and its effect of nuclear plasticity has been shown by our group. And we were also interested in studying the different biophysical aspects of the lamin association, the kinetics of association, the thermodynamics of association. We did extensive isothermal titration calorimetry experiments, which I think was done first by us. We did quite a lot of single molecular force spectroscopy to address the individual elastic responses from the different domains of the lamin. Because as you understand, lamin is a big protein. So all these things were actually very, very interesting, very fascinating, and we had published it. We had a lot of citations also till now. And eventually, we also were interested in finding out the effect of mechanotransduction from the extracellular matrix to the nucleus, and particularly in the presence of mutant lamins. And that actually we do more or less based on a kind of tissue engineering approach, where we modify the matrix stiffness and also the topography and see how the force is transmitted to the nucleus when the nucleus contains the mutant lamin vis-a-vis the wild-type lamin. The disease model has been muscular dystrophy. So I started my lab by studying the dilated cardiomyopathy, as there are a lot of patients suffering from the dilated cardiomyopathy in the area from where I come from, the southeastern part of India, the eastern part of India. And also we have started studying the role of the DNA damage repair mechanisms, which have been earlier hinted upon. And we went a bit further, and we devised the lamin as the diagnostic markers, and we actually patented that. We are in the process of patenting that based on the machine learning techniques, nuclear morphology based machine learning techniques. So this part will be talked about by Duhita, and the viscoelastic part and some of the structural analysis and the muscle differentiation, effect on muscle differentiation will be talked by Chandrayee subsequently. So just to give a brief overview, this is the expression of the lamins in the cancer. So when I say that I studied the two different diseases, this has a very important meaning in the sense that in the cancer, to date, there are no mutations, right? So it is the level of expression of lamin, which is either upregulated or downregulated in the different types of cancer, what you can see here. We are primarily interested in this ovarian cancer, ovarian serous cancer, where we will explain our results, where actually Duhita will explain her results. And in the case of, so this is basically the biology of the ovary and the ovarian, the surface epithelial cells. I will not go into the details. And the second aspect of my research is the disease or the diseases associated with the lamin A mutations, which are close to 500 something now, which are associated with the missense mutations of LMNA, and which lead to the diverse array of diseases, which are termed as laminopathies. My lab studies mainly the dilated cardiomyopathy and Emery-Dreifuss muscular dystrophy. We have generated 28 mutations. We have reported most of these mutations based on their biophysical studies and some of their cell biological properties also. And many more interesting things are on the platter. So as you know, in the dilated cardiomyopathy, the understanding the role of the elasticity or the viscoelastic response of lamin is very important. And that was the rationale of beginning of the viscoelastic studies, because with each systolic diastolic cycle of the heart, there is this, this expansion contraction of these ventricles, and in the DCM we see an enlarged ventricle, left ventricle, large ventricle, both the ventricles, which leads to the cardiac arrhythmias, and if untreated, a large number of patients in India are untreated, and this ultimately leads to myocardial infarction and death. So basically what happens here is actually the lamins in the nucleus of the myocytes, which are the cardiomyocytes lining this in the myocardium, actually, they lose, or kind of elasticity, I would say, sacrificed to a certain extent, so that the ventricle, the volume doesn't come back to the same state after each systole diastole cycle. And as a result, we see an progressively enlarged ventricle. So this was the genesis. So these are some of the facts about lamin, which I think most of us know by now. So the structures of the lamins, the Cax box, where it is phenycylated. So the important take-home message from here is B1, B2 maintain the phenylate group, where A and C, of course, C doesn't have the phenylation. A is phenycylated and then loses it. And to the right, there is this famous model, which has been also reviewed by Nico Stuman, 1998, shows the schematic organization of the lamin, starting from the monomers to the dimers to all these different forms. And actually, it is here that we address the different problems, that is by the biophysical experiments, where we could identify how these rate-limiting steps of formation of the dimers were seriously inhibited, which ultimately led to misshapen the higher order oligomer formation on network. So our hypothesis for the lamin A mutation-based studies, that is the cardiomyopathy and the EDMD, lay in the fact that we chose to address the pathways. One is the lamin A mutation leads to mechanical changes due to the deformed lamina, which we have all seen, which most of us have reported in a consensus manner. And lamin A also leads to a transformed epigenetic landscape, which ultimately leads to an altered gene regulation, and this leads to disease phenotype. So we wanted to reconcile these two hypotheses to actually build up the cause of the pathophysiology of the dilated cardiomyopathy primarily, and later on EDMD. So these are some of the papers in my lab which have talked about mainly viscoelasticity and the elastic responses, which made very important strides in the field. So what we could summarize just in one slide, will not take much time, is that evolutionarily why we could explain kind of from our studies that why A is actually switched on at the later stages of differentiation, or let's say at when the B is, or why B is primarily there and A comes next. So we could actually reason that B actually could resist the strain deformation up to a certain degree, after which you remember the figure from Goldberg, that there are the two arrays. One is the orthogonal array of fibers overlaid by the parallel array of fibers, so which is actually overlaid on the B, which can take care of more of increasing the stress mechanical cues in general when the threshold limit is reached. So I would not prolong my talk. So this is the city of Calcutta. This is the lifeblood, the Ganges. It's a beautiful city with this the cricket stadium, which is the lifeblood of India. We play a lot of cricket. It's a beautiful city. All of you are welcome to visit Calcutta, and also the Saha Institute of Nuclear Physics, if you can arrange some international meetings here. Subsequently, you would hear, I just changed the order of the talk. So it will be first Chandrayee, who would be talking about these EDMD mutations associated with EDMD, associated with lamin A mutations, excuse me. And then it will be Duhita, who would be switching gears and taking you on a ride to the ovarian cancer. So it's Chandrayee, the podium is yours. Thank you.
Okay, thank you. My name is Chandrayee, and Firstly, I would like to thank the organizers of this meeting and Sharka for providing me this platform to present my talk today, and to my supervisor for introducing me. And today I'm going to talk about effects of dilated cardiomyopathy and EDMD mutants of lamin A in on nuclear architecture and function. Okay, so as Dr. Sengupta introduced, our lab for the last decade has been dedicated actively towards studying the root of pathogenesis in these laminopathic cells, and we have generated to that end around 28 mutations in the protein until now, and we have extensively studied those proteins in vitro and ex vivo. So when a lamin protein is mutated, it is structurally and functionally compromised. A change in the secondary structure induces a change in the higher order structure formation, which gives rise to a change, an alteration in its viscoelastic property. Our lab has already studied the viscoelastic property of lamin A proteins, both as a bulk property with the help of rheometers and as single molecules by the elasticity of single molecules by domain-specific study of single molecule force spectroscopy. And as an extension to that study, I have tried to determine the viscoelastic properties of lamin with the help of optical tweezers, which is a parallel high throughput process, which is much more faster and a robust process than the commercially available rheometers. So in this process, we have a probe, which is a bead in this case, which is perturbed by an external force, which is a laser in this case. And by this, we can measure the frequency dependent complex viscoelastic properties of proteins in solution. And this is the setup of the instrument that we have used in our study. So for my study, I have purified the full length lamin A protein, both the wild type and the mutant, in a bacterial expression system. And the mutant that I have worked with in my first project is A350P. There is a proline in place of alanine in the 350th position of the protein. So this protein is like this mutation is the rationale of choosing this mutation is based on the severity of phenotypes that we have seen in patients that have been reported. And we know that insertion of a proline in the alpha helix inserts a bend or a kink and destabilizes the structure. So we thought that this would give us significant structural alteration if we study the viscoelastic property of this protein. So initially, we started with the measurement of secondary and tertiary structural alterations by CD spectroscopic studies and fluorescence spectroscopic studies. So when we studied the viscoelastic properties of the proteins, we found similar observations for wild type as we previously observed. The lab had previously observed for the wild type protein. But interestingly, for the mutant protein, we saw higher storage and loss modulus for the mutant protein, particularly at higher concentrations. So what does it mean? It means that the mutant protein is probably forming a much more compact, denser network. So to corroborate that, we stained the protein with antibodies. We plated the proteins in vitro on previously coated fibronectin cover slips, and we imaged the proteins under super resolution microscopy. And indeed, we saw an increase in the network, increase in the compactness in the network formation, and we saw smaller mesh size. So now we thought, what must be happening in the cell? So if this protein is forming a much more denser network, so is it making the nucleus much more stronger than the wild type? So to answer our questions, we We cloned this particular mutant under EGFP vector and transfected the mutant in human myoblast cells. And what we saw was really interesting, because the intact lamin rim for wild type was completely missing in case of the mutant. And we saw that the mutant was forming a much more denser like aggregates in the nucleoplasm. And a closer look at this aggregates told us that the mesh size was also smaller, and it appears that this aggregate is forming at the cost of the rim. And this is also reminiscent of the higher G prime, G double prime values that we obtained from our viscoelastic measurements. And maybe it's turning the protein structurally and functionally like it's compromised. We have already published this part of our study in 2021. And as a summary, we saw the increase in storage and loss moduli for the mutant protein, and it entailed a smaller mesh size, and higher erratic mesh density implied lower elastic response for the A350P to implied stress that might even lead to catastrophic failure on the application of localized stresses on the nucleus. For the next part of my study, I have investigated the IG fold domain of the protein and some mutations associated with the EDMD disease. So this IG fold domain harbors the most deleterious mutations in the protein, and our lab incidentally have already established the with some other EDMD mutations that this involvement of IG fold domain in the structural alteration of the protein. And as an extension, I have tried to find the residue specific fluctuation that these mutations are causing inside the protein, and as a whole, how the whole protein dynamics is changing inside the cell nucleoplasm. And to study that, we conducted a fluorescence correlation spectroscopy to study the protein dynamics in the single molecule level. And we studied with 3 mutations here, R453W, W498C, and R. And we found lowest diffusivity values for R453W. That means that this protein is taking the longest time for diffusion. So to connect our observation to our previous like the observations from the lab, we had already seen that R453W mutation in the IG fold domain caused the domain to open up, because it breaks a very important salt bridge between two glutamate residues at 443 and 444 position. And so it gives the molecule a much more voluminous structure. So that might be causing the protein to take a longer diffusion time across the cell. So to support this observation, we conducted a molecular dynamics simulation on the IG fold domains, where we saw different electrostatic surface potentials on the mutants in comparison to the wild type. And when we wanted to study the stability of the protein, we with the help of RMSD and RMSF studies, we saw that in case of R453W, there are in certain residues, there are fluctuations which are not noted in the other mutants. So so in this case, the residues near the mutations, the eigenvectors show the direction and the magnitude of the fluctuations of this residue, which is not noted in the other mutants or the wild type protein. We also studied the altered secondary structure for the mutant proteins with the help of DSSP tool. And as a summary of this work, we obtained smaller diffusion coefficient and longer diffusion times for R453 as compared to wild type and 498 C and R. We interpreted this result as a contribution of motion for a more voluminous molecule with longer diffusion times, and IG domain exhibits faster stochastic movements leading to a higher preponderance of random self-association to form aggregates, thereby lowering the probability of a stepwise steady association into a functional nuclear lamina that we see in the wild type proteins. In the next part of my project, I wanted to see now how these laminopathic cells they behave in presence of compressive stress. So to recall the route of mechanotransduction, when a cell experiences stress in terms of stretching or like compressive force, the cues are transmitted inside the cell cytoplasm by the receptors through the cellular microenvironment containing these proteins, mechanosensing molecules like actins, microtubules, which apply differential mechanical force on the nucleus, which in turn takes these signals to the chromatin through the major mechanosensing molecules of the nucleus, which is the lamins, and thereby causing alternate gene expression or DNA condensation. So I wanted to see now that in the laminopathic cells, when the nucleus is compromised because of the lamin mutations, how these other molecules in the pathway of mechanotransduction, they react to this compressive force. So we conducted a very simplistic experiment to that extent. We placed a weight on the cells and removed for some time and removed the weights and then allowed the cells to recover, and then stained the cells with actin proteins. And here, interestingly, what I saw is that the actins, in case of the mutations, showed a more branched structure. So to see whether the alpha actinin, because it is a branching inducer, how the alpha actinin is expressed or localized in the cell, I stained the cells with alpha actinin and also the Nesprins and the lamin B. I did not see much change in the lamin B because it did not localize with the lamin A aggregates, but we definitely saw Nesprins to have a different localization than the wild type. So in this part of the project, these are some very preliminary studies. I need to like see the cells under the super resolution microscopy for a better understanding. We have already sent the samples for RNA-seq, and we are expecting the results so that I can come up with much better understanding of why these phenotypes are observed. So these are my conclusions and these are my publications, and we have filed a patent for the optical tweezers work, and this is my lab. I would like to thank my supervisor, our collaborators, my lab mates, my friends, and thank you for your attention. I would like to take questions.
Hello, everyone. I would first like to thank the organizers and the coordinators of Connecting Intermediate Filament team, and would like to thank my supervisor for introducing me. So I would be talking about the role of lamin A in DNA damage response in ovarian cancer. Now, so nuclear lamins are the potent regulatory factors which, excuse me, sir, can you please turn the speaker off? It's all right. So, as I was saying, that nuclear lamins are potent regulatory factors to assess the potential of the cell towards carcinogenesis. It alters the cell's nuclear mechanomorphological features, which are very potent regulatory factors for assessing the malignant potential of a cancer cell. So before going to the entire study, I would like to let you all know that the entire study is based upon ovarian cancer, where we can see the modulation of the nucleatia is associated with changes in nuclear morphology and nuclear function. So the entire project has been subdivided in these two major parts, where we will be talking about the altered nuclear morphology with respect to the differential expression and localization of nuclear lamins, and concerning the change in the nuclear function. So the first part deals with the change in the nuclear morphology. So the project started by staining patient tissue samples from ovarian cancer patient tissue samples, which we derived from Tata Medical Center, Kolkata. We stained them with lamin A and B. And what was the most prominent and consistent observation, that all the nuclei were much bigger in size for the cancerous tissues with respect to the normal tissues. And we, after staining all those samples, we generated two types of input data from them. One was the numerical values from the morphometric parameters calculated from the nuclear morphology, and another one was the lamin A and B stained tissue images as a whole. So the first pipeline took into account the numerical values from the morphometric parameter, and that was fed into the system, and which gave us a particular probability score. And the second pipeline took into account the pre-processed images of lamin A and B stained cancer and normal tissues. And that also gave a particular probability score, and the maximum probability score from these two pipelines gave us the decision whether the tissue is of cancer or normal type. Now, regarding the first pipeline, we calculated all the morphometric parameters. We calculated 8 total parameters, within which area and perimeter values were most significantly altered. And we could see that most number of nuclei in the cancer tissues are of 55 to 62% higher area than the normal nuclei. And the similar observation was that for the perimeter value as well. And regarding the second pipeline, the images went through steps of pre-processing, where the pre-processing operations were designed such that the key visual properties of the nuclei gets more illuminated. So after this two pipelines and feeding the input data into these two pipelines, we got a particular decision regarding whether the tissue is of cancer or normal type. And we could see that this algorithm is able to classify ovarian cancer versus normal tissue with 99.8% training accuracy and 99.7% validation accuracy. And we also had a different set of test data set, which we used for testing our algorithm. And while testing, we also checked the performance of several other combination of deep learning networks, but we could see this sort of deep hybrid learners, which take into account both conventional neural network and machine learning classifiers, are the best model for the specific problem to classify ovarian cancer. Now, these are the conclusions from this project, and we are in the process of filing a patent regarding this. And the next project that I was just mentioning, that the nuclear functions are also altered based on the expression upregulation of lamin A in ovarian serous cancer. And the nuclear function which will be talked about here is the DNA damage repair response. So in this case, from the literature studies, this has already been reported that a system, when it is having lower expression of lamin A, it will lead to higher nuclear curvature, which in turn will lead to increased rupture and excess DNA damage. But when the system will be having higher expression of lamin A, that will result in increased mechanical stability and better protection to the chromatin, which will result in higher chemoresistance. Not only that, when a system will be having higher expression of lamin A, that will result in increased stability of the repair proteins, which in turn can result in higher chemoresistance. So this project has been worked on two different cell lines, OFCA3 and IOAC. OFCA3 is ovarian serous carcinoma cell line, and IOAC is immortalized ovarian surface epithelial cells, which are the control cell line for the study based on the epithelial origin of ovarian serous cancer. Now, first of all, in these two cell lines, we checked the expression and distribution of lamin A and B and could see that there is prominently higher expression of lamin A with respect to the normal cell, and also the nuclei size are bigger. And we checked the status of DNA damage repair in these two cell lines and could see that the homologous recombination mediators are already endogenously elevated in the OFCA3 cells. Now, to see how the DNA damage repair mechanism works in a condition in ovarian cancer where there is already higher expression of endogenous lamin A, for that we treated OFCA3 cells with etoposide, which is a topoisomerase 2 inhibitor, and the DNA damage was validated by COMET assay and also checking the expression of gamma H2X and PCNA. So after that, we started the entire or the global gene expression after treating OFCA3 cells with etoposide, and could see that 205 genes get differentially expressed, which majorly belonged to the pathways like double-strand repair, non-homologous end joining, telomere organization, DNA packaging, etc. But what was most interesting is that in addition to these pathways which were already expected, we also got some of the genes from pathways like PI3 AKT, TGF-beta, and genes like BIRC3, ID3, BIF1, which were indicating a sort of chemoresistance in the system. So we selected 12 such genes which are directly associated to cellular proliferation, chemoresistance, and apoptotic evasion, and revalidated their expression by qRT-PCR, and wanted to see whether this upregulation of these genes in the etoposide-treated OFCA3 is specifically due to the expression upregulation of lamin A or not. We knocked down lamin A in the OFCA3 cells and checked their expression again, and could see that those gene expressions are all downregulated except for PLK1. After that, we also treated those LMNA knockdown OFCA3 cells with etoposide and could see those gene expressions which were upregulated in the normal OFCA3 cells are all downregulated in LMNA knockdown cells, which were actually indicating that lamin A has direct or indirect roles in the gene expression of those genes associated to cellular proliferation, apoptosis evasion, or chemoresistance. To further go into to further know the molecular orchestration behind this entire event, we did a sort of bioinformatic exercise by analyzing the lamin A interactome and could see 5 of the genes from the 12 genes under study were direct interactors of lamin A. And we also searched for their transcription factors and could find that 12 such transcription factors which regulate those gene expressions are direct interactors of lamin A. We also could find that these transcription factors, 4 major transcription factors, which bind to the promoters of those genes, are downregulated in the LMNA knockdown etoposide-treated OFCA3 cells. So this all information or observations are suggesting that when a system of ovarian cancer cell with high endogenous level of lamin A is experiencing a sort of DNA damage in form of etoposide or any chemotherapeutic agent, it is indicating emergence of chemoresistance, apoptosis evasion, and proliferation, but that is not the phenomena when a cell is having lower expression of lamin A. So these are the conclusions from the project. And I would like to briefly explain another project which does not deal with cancer, but with laminopathy. I started another laminopathic mutation called K97E, which is a very deleterious mutation causing dilated cardiomyopathy. And this is a very preliminary data that I want to share, that we have checked the expression of the epigenetic modifications in the cells after transfecting it with a laminopathic mutation called K97E, and could see that there is a reduction in the heterochromatin, facultative heterochromatin content of the cell in form of loss of H3K27ME3 mark, and also could check the gene expression profile there and could see that those genes were differentially expressed majorly, which are all linked to inflammation pathways like kine and interferon related pathways. So right now we are actually performing qPCR experiment to link whether this epigenetic alteration, modification alteration is actually causing this sort of changes in gene expression in the inflammatory pathways. So these are the list of my publication, and I would like to acknowledge my supervisor, the coordinators of the meeting, our collaborators, and friends, parents, and family. Thank you.